Half of my colleagues are not here. Morning. So, not really. Left is forward, right is back, and that's it. You'll get there'll be a flashing yellow light for when you have 5 minutes to go, and then it'll start flashing when you have 2 minutes or something like that, and then it'll turn red. Okay. Please find your seats. We're about to begin. Good morning, everybody. I hope you had a nice evening and are ready to take on another day. My name is Birte Nielsen. I'm the Research Director at UFAW, and I am chairing this morning's session. For those of you who were not here yesterday, I still have some practicalities. In the event of a fire alarm, there is not a practice scheduled, so if the fire alarm goes off, please leave the building and we will gather outside 14 Queen Street. The tea and coffee and lunch are served as yesterday. It is vegetarian but with vegan options. And if you have special dietary requests, please speak directly to the catering staff because that will have been prepared for you separately. There's not a drinks reception tonight because this slide is from yesterday, so please ignore that bit. To access the Wi-Fi, this is the network name and the password. It will also be constantly on the side monitors here. There are still photographers and videographers that are filming, and if you want an attendance certificate, you can pick one up at the reception desk. This is the hashtag for use if you want to tweet from the conference. I could see that the Twitter was quite alive yesterday. And the posters today, they are still up, and if you could please attend your poster after lunch and then take it down before 5 p.m. today. We are running a poster competition, and that will be announced at the closing of the conference in here after the two workshops and debate forum. I think it's at 10 past 5. Yes, so the debate forum and the workshop is taking place today at 20 past 2. The workshop will take place in here, and we have the capacity of the theatre. The debate forum, which is about animal welfare labelling, is now full because the room can only take 60 people. So the people who can get in will have a black dot on their badge, and please don't find a Sharpie and make one because I just gave that away, didn't I? And I will just finish off with this slide again. Hugh mentioned it yesterday, that we are funded solely by donations and legacies from members and supporters. And the reason why I want to put up this slide again today is linked with the announcement yesterday that our journal, Animal Welfare, is flipping to, it's a technical term, but flipping to full open access online publication with an article processing charge. And one of the things that Hugh mentioned was that if you are from an institute where you have a transformative agreement with our new publisher, Cambridge University Press, you do not have to pay anything. And if you don't have the money to pay for it, you can apply for waivers. One thing I want to add to that is that if you are a member of UFAW and you have to pay the APC, you will get a 20% discount. And if you are a member of UFAW, you will also get access to our back catalogue. All our previous issues of the journal will be open access online for people who are members of UFAW and for libraries that buy the access to them. So I would also like to encourage you, if your institution do not have a transformative agreement with Cambridge University Press, to perhaps speak to your librarian and say, why don't we have that? Because then I can publish for free in Animal Welfare. So thank you for that. And with that, I will put on my chairing hat and introduce the first speaker of today. This is Professor of Ethology, Melissa Bateson. She's also Director of the Centre for Behaviour and Evolution. And she has brought with her, she has just recently published the 4th edition of the book Measuring Behaviour. It used to be written by her father, Sir Patrick Bateson and Paul Martin, and now she has done the 4th edition, and I can say it's excellent. And she sounds like a sort of clandestine business. She'll sell it to you for £20, but if you pay cash, yeah. But she only has a limited number, so seek out Melissa, and she'll do the deal. And with that, I would like to welcome Melissa Bateson and take it away. Thank you.
Thank you, Birte, for the invitation to give this talk today. I was asked by UFAW to address the use and misuse of welfare indicators, and I spent some time kind of racking my brains trying to decide what I should talk about, and decided to focus on body weight for reasons that will become clear. So for much of my career, I've worked on this small bird that you can see in the middle of this photo, the European starling. So for my PhD, I studied foraging decisions in these birds. And in the last decade or so, I've become really interested in using starlings as a model in which to understand regulation of body weight, possibly with relevance to understanding the obesity epidemic in humans, hence this rather nice picture of starlings with humans. I think starlings are a really nice model for understanding this problem. They are like us in many ways, despite being birds. They're incredibly adaptable and have followed us around the world and live in many of the same environments that we do, and even enjoy fish and chips on Tyneside, as you can see here. Okay, so there's huge variation in the body weight of apparently healthy starlings kept in identical conditions in our lab. On the left here, you see a graph of masses of a group of our starlings, and you can see that they vary from 71 grams to nearly 100 grams. These birds are living in the same aviary, eating the same food. They come from the same population in Northumberland. And in fact, these birds come from just 8 nests, so many of them are full siblings. The only difference between these birds is that 10 days in their early development when we manipulated various aspects of their feeding. But what you can see here is this massive weight range. If we look at the residuals, when we take their skeletal size into account, you can see that even then, the residuals range from minus 10 grams to plus 15 grams. So this really is a massive range that we need to understand. And I should say that these birds were apparently healthy and lived in our lab for many years after the period when we reared them. So how can we understand some of this variation? Well, so our research has shown a number of different factors, and obviously there are many other factors studied by other people. So we know that genes, epigenetic inheritance are important from studies on humans in particular, and also rats. I've just shown you that skeletal size is likely to be important. In starlings, we know that sex is important. Males are slightly heavier than females typically. My own research has shown a big influence of developmental history. So we know that the feeding regimes experienced by these birds in the first few days after hatching has a huge effect on their adult body weight. The birds also change weight massively across the day. So they gain about 10% of their body weight between dawn and dusk each day and then lose it again each night. There's also a big effect of time of year, which can be between 10 and 20%. There are effects of diet, which we haven't explicitly studied, and of the current environment, and I'll talk about some of these later in the talk. So there's a huge number of sources of variation in weight that we need to understand in animals in general and in these birds. So what I want to talk about today is body weight as a welfare indicator. So in the course of my research, I've come up against the problem that the Home Office doesn't like loss of body weight in animals and believes that it really is a sign of poor welfare. And as I've just shown you, we have evidence that our birds are varying massively in weight by as much as 20% in birds that seem to be apparently healthy, or maybe they're not. But this has been something I've had to contend with in my research, in getting project licenses to conduct my research, to try and defend the idea that we can ethically study some of this variation in body weight without causing enormous stress to these animals. So what I want to talk about today is whether or not body weight is a good welfare indicator, and whether we ought to be worried about starlings that are losing 10% of their body weight. You can see that in rodents, a 10% loss over a week is regarded as mild severity, between 10 and 20% moderate severity, and over 20% would be severe, and would often be regarded as a definition for a humane endpoint in rodents. So we have had our starlings lose 20% of their body weight in my lab, and show no other signs of ill health. So I really didn't want to have to kill birds that are apparently healthy. So I really needed to kind of understand what the relationship is between body weight and welfare, and whether this is a reasonable indicator of poor welfare that should actually cause us to kill an apparently healthy looking bird for no other reason than that it's lost 20% of its body weight. So there is quite a good precedent for using body weight as a welfare indicator, even in the literature on birds. So this is a 2022 paper that I found the other day, which is developing a new plumage-based score for zebra finches as an indicator of well-being. And it actually uses fat condition scores, which are derived from body weight, as a gold standard for comparing these new plumage condition scores against. So it's not only regarded as an indicator of welfare, but actually as a gold standard in some recent studies. There seems to be a general assumption that fatter equals healthier equals happier. So I just reproduced a sentence from the introduction of this zebra finch paper. They argue wild-caught animals may suffer from stress in captivity and lose weight, and environmental stress in poultry may reduce food intake and thus impair weight gain and reduce fat reserves. So this is just an indication of the kind of arguments that you're seeing in some of the veterinary literature for the relationship between weight loss or weight gain and welfare. So clearly body weight has a lot of advantages as a welfare indicator. It's objective. We can measure it accurately and repeatably, so it has good intra and inter-observer reliability, which ought to be a good indicator for a welfare indicator. It's fast and cheap to obtain, and it's relatively non-invasive. But is it a valid welfare indicator? All of these features are good features, but they're not sufficient for us to have a valid welfare indicator. In thinking about this, I think it's useful to think about the kinds of errors we can make when we use any welfare indicator. So what I've shown here is a group of birds, and the decision that we need to try and make about these birds is, do they have poor welfare or good welfare? So we want to divide the birds into birds with poor welfare and birds with good welfare. So that's this vertical line down the middle of the slide here. We can draw a circle which describes the group of birds within our population that are the animals losing body weight. So say we have 10% loss of body weight, and these are all the birds inside this grey circle. We can then ask, how does that indicator relate to the true welfare of the birds, so whether they have poor welfare or good welfare. And we can divide up the birds into whether they're what we're going to call false negatives, so birds with poor welfare that we're missing with this indicator, true positives, which are the birds that have lost weight and indeed have poor welfare. We've got the false positives, which are the birds that have not lost weight and have that have lost weight but have good welfare. And then we've got the true negatives, which are the birds that have good welfare and haven't lost weight. So we need to worry about the errors that we can make with any kind of welfare indicator. And we normally ask two sorts of questions about these kinds of data. So one relates to what we call sensitivity, which describes the probability that weight loss allows us to diagnose a welfare problem correctly. So in my previous diagram, that's these true positive birds here. So sensitivity is just the ratio of the number of true positives to the total birds with poor welfare. So clearly, the higher this number, the more sensitive our indicator is for picking up cases of poor welfare. So that's true positives divided by true positives plus false negatives. The other thing we want to ask about is specificity. So this is there's a typo on this slide, that should say specificity. Describes the probability that normal body weight allows us to infer good welfare correctly. So going back to my picture, that's really these birds here. So these are the true negatives. So what we're calculating here is the number of true negatives divided by the total birds with good welfare, which is the true negatives plus the false positives. So these two measures, sensitivity and specificity, give us some sense of how well our welfare indicator is working in discriminating poor welfare from good welfare. Okay, so having got that bit of theory out of the way, we can ask whether body weight is a sensitive and specific indicator of welfare. How often does it lead to false positive and false negative inferences about welfare? And I think to answer this question, we need to understand the reasons for weight change in animals. And I think we can broadly divide these into two categories. So we can have pathology, which is due to starvation or underlying disease. And I think this is what underlies the adoption of weight change, weight loss as an indicator by much of the veterinary community. It's clear that if animals are really sick or if their environment is totally inadequate and they don't have enough to eat, they will lose weight, and that is a bad thing, and we can call that pathology. But what we need to understand is that there's also a lot of adaptive plasticity in body weight in animals. And I'm going to talk specifically about adaptive plasticity due to strategic adjustment of the starvation-predation trade-off, which is something that's been discussed specifically in the context of small birds, but actually the theory underlying this could apply to any animals, and I've recently been arguing that it could apply to humans. The logic in the models is not specific to small birds, and I think there's no reason to believe that these sort of arguments couldn't also apply in many of our other laboratory animals, such as rodents. So just to give you a little bit of insight into the theory here, the idea is that fat has both costs and benefits. So we tend to think of the benefit of fat very clearly as being protecting animals against the risk of starvation. So if we plot the probability of starvation on the left-hand y-axis here, probability of starvation reduces as body fat increases. So that's a benefit of fat. But fat also has costs, and the one that the bird people usually think about is probability of predation. So the idea is that as you get fatter, you become less agile and less able to avoid predators. So actually, as body fat goes up, probability of predation also increases. So there are these two conflicting demands on birds in terms of how much fat they should carry. So just to convince you that in our starlings, having more fat does actually impair their escape flight performance, I just thought I would show you this data from an experiment done by one of our students, Dan O'Hagan, a few years ago for his master's project. What Dan did was to have starlings sitting on a perch near the floor in an aviary. He then played them using a doorbell that we bought in B&Q, the sound of a barking dog. So it was a remote button that you could press in the room outside and scare the bird with the sound of a barking dog. And the bird would take off and fly up to this far perch on the other side of the aviary. It had to fly through a maze of an aerial maze of strings, because we were going to look at whether it hit the strings as one measure of its agility. In fact, we found no effects on string hitting, but what we did find was that the takeoff speed of these birds went down as their body weight went up, and also the takeoff angle went down as their body weight went up. And I think you can see that if you take off slower and more shallowly, you're much more likely to be caught by a terrestrial predator such as a cat. So I think this is some nice support for the idea that having more fat might actually have an effect on the survival of these birds. There are many other such experiments that have been done. This is just one from our lab supporting this general idea. Okay, so given that we have costs and benefits of fat, how much fat should animals carry? Well, the optimality model suggests that optimum fatness will be where you minimise the joint costs of the probability of starvation and the probability of predation. So that would be where these two lines cross, which would be this point on this graph here. So that's the optimum fat to carry given these two competing demands that the animals have. If predation risk, for example, increases in the environment, then that will change the shape of the probability of predation curve, and that will then shift optimum fatness downwards. So if there's higher perceived predation risk in the environment, animals should lose weight. Similarly, if there's increased starvation risk, the benefits of fat will be higher. This curve will shift upwards, and optimum fatness will shift to the right and increase. So we can expect the fat that birds carry to reflect this trade-off between probability of starvation and predation. So the predictions arising from this theory are that animals should possess evolved psychological mechanisms for sensing and responding to cues of possible or actual starvation risk, and this might be things like food insecurity in the environment, or perhaps social dominance, which allows you to outcompete other individuals in your group for food, and also possible or actual predation risk. And there's a lot of data actually backing up these predictions from the optimality models. So I want to show you some data from an experiment done by Claire Andrews. What we did here was we manipulated food insecurity, which we did by taking away the bird's food for 5 hours, a continuous period of 5 hours at an unpredictable time of day on 5 days of the week. And we did this for a period of 19 weeks. And you can see we had the insecure birds, which are the red line here, and the control birds, which had ad libitum food the whole time in their aviary. And you can see that over the course of this 19-week period, the insecure birds always had heavier body masses as predicted by the models compared to the control birds that had ad libitum food. The absolute masses of the birds went up and down with time. This was quite a long experiment that took place over the winter. Temperature was changing, which probably affected body mass. So there are absolute changes, but the point is that the blue line of the control birds is always below the red line of the insecure birds. And at the end of this 19 weeks, the furcular fat scores, which you can see visually on a bird, were higher in the insecure birds compared to the control birds, supporting the idea that this is not some change in muscle or something, it's a change in fat reserves in these birds. So this is a case where doing something that you might expect would reduce the welfare of the birds apparently also increases their body fat. In this experiment, we also measured various other more validated measures of welfare. So I've just shown the two measures here. One was feather regrowth. So we pulled a tail feather from the birds at 3 points during this study. And one of the things we found was that in the insecure birds, their tail feathers regrew a bit slower and a bit less long. So that suggests that there was some kind of impact on the insecure birds in that they were investing less energy in somatic maintenance and repair. The other piece of evidence we have is that the erythrocyte telomere length, which is the length of the telomeres, which are the caps on the ends of the chromosomes in the red blood cells of the birds, decreased in the insecure group. And telomere length is, I think, an indicator of exposure to stress that is really quite well validated across a number of species now. And we've been exploring it as a measure of stress exposure in the starlings over the last decade, and have shown that it does seem to respond to stress experienced over various periods of life. So I think this is kind of quite compelling evidence that although these insecure birds were putting on fat, they were also showing evidence of poor welfare as indicated by having shorter telomeres, which actually predicts that they will live less long. So they are predicted to live less long as a result of this marker of accelerated biological aging that they have. So here we've got kind of validated welfare indicator that is lower in the birds that are actually fatter. And we think there's actually a causal relationship here. So the food insecure birds did not actually eat more food, interestingly, over the course of this study. We think that they paid for their increased fat reserves by diverting energy from somatic maintenance and repair, so repairing their telomeres and feathers, into fat reserves. So that's how they created their higher fat reserves. So I think this is a really interesting example of how understanding the biology of what underpins changes in body fat is really critical to understanding whether it could be a good welfare indicator or not. And I think this really suggests not. I also wanted to show you some data from another experiment. This is one done by Mark Witter and John Swaddle back in the mid-90s. So in this experiment, they manipulated dominance hierarchies in small groups of starlings. So they had groups of 3 birds, and they measured the dominance in the group by looking at who outcompeted who for access to a food bowl. And what they found, which is a common result, is that the most dominant birds were actually the lightest, and the least dominant birds in the group were the heaviest. So you tend to get this relationship in birds. And this is probably comes back to access to food and food insecurity again, that if you're more dominant, you can have food when you want it, and so your probability of starvation is going to be lower compared to a subordinate bird that can always be pushed off the food bowl. Anyway, what they did in these groups is they did an experimental manipulation where they removed the most dominant bird or the least dominant bird from the group, and then looked at what happened to the body weights of the remaining birds. And what they found is that if you remove the most dominant bird, the remaining 2 birds in the group, 3 and 2 in this case, actually lose weight. So that's a demonstration that the position in the dominance hierarchy is actually causal in changing the body weight of the birds. So in this case, an increase in dominance, which you might think might be a positive thing for a bird because it's reducing probability of starvation, is causing a decrease in body weight. The final experiment I wanted to show you is one where perceived predation risk was manipulated. This was actually in great tits, and here you can see graphs of great tits where they were either in a safe environment with no sparrowhawk, or there was a model sparrowhawk present. And these are within subjects data showing the same bird in the 2 different conditions of safe and predator. And you can see that when the predator was present, the grey bars, the birds were always lighter. So this is another nice example of how birds can change their weight in relation to perceived stress, in this case predation risk, and in this case their weight goes down. So just to summarise these different effects, I've shown you that different types of stress can have opposite effects on body weight. So food insecurity leads to weight gain, dominance leads to weight loss, and predation risk leads to weight loss. So there's no simple relationship between change in weight and the likely valence of the experience of the birds. I would argue that food insecurity is probably, if anything, stressful in an unpleasant way. Dominance may be more likely to be positive and certainly associated with reduced food insecurity. So I think it relates to food insecurity. Predation risk is likely to be something that's frightening, so induces greater anxiety in the birds, and that does lead to weight loss. But there's no simple relationship here. Therefore, I think that weight loss is not really either a sensitive nor a specific welfare indicator. We probably shouldn't use it, in birds at least, based on these data, as a decision-making tool, especially for humane endpoints. I think that seems crazy given what we know about the plasticity of bird body weights. Okay. So this is a graph that I have to thank Georgia Mason for encouraging me to think about in the context of deciding whether welfare indicators are good or not. So what I'm plotting here is the likely valence of the animal's experience. So this is the welfare axis going from negative on the left to positive on the right, and body weight on the y-axis here. And if I had to think about what the likely relationship might be in starlings and probably many other species, I think it's likely that very low body weight is probably associated with poor welfare. We know that before animals starve, they're going to have poor body weight. And we've seen animals get sick and die where their body weights have plummeted. It's also likely that very high body weight's associated with poor welfare. It's associated with poor health, and it might be associated with animals struggling to move and being more worried about predation, less able to do lots of things. So it seems likely that there'll be a non-linear relationship here. But it also looks like there's a very wide range of weights where the welfare might be similar, and where small changes in either direction don't mean increases or decreases in welfare. So I think there's quite a sort of flat top to this curve here, side to this curve, which means that it's really not going to be a very useful welfare indicator a lot of the time. There might be parts of the curve where it could be useful, but I think in general we have to be very dubious about whether body weight is going to tell us much useful on its own about the welfare of animals. So I just want to finish off with something a little bit different that's come out of my work. It's related, but not quite the same. So something that's really bothered me about measuring body weight in the starlings is that I think it's probably acutely very stressful for them. So what you can see here is one of our starlings being weighed in the way that we always used to do it. So what we would do is catch the birds from the aviary, which is a stressful business. It normally involves turning off the lights, going in there, grabbing the birds. We do it in the dark because that seems to be the easiest way to get them without them flying around too much. We grab the birds in the dark, then they're put into a plastic cone here. I think this is actually a decapitation cone. We don't decapitate the birds, but we put them in these cones to weigh them. There's a hole in the end here, and it's just a convenient way of restraining a starling. So you can see this bird sitting on the scales here. We know from lots of other work that catching and restraint in small birds is an acute stressor, causes a massive spike in corticosterone that takes a couple of hours to go back to normal. So this is an acutely stressful experience, and if you're doing this on a regular basis, the birds are likely to get pretty stressed. So one of the things that I've been trying to do in thinking about how to study body weight in the birds in a more ecologically valid way, without stressing them the whole time by catching them to weigh them in this way, is to develop a system where I can actually weigh the birds automatically. So this is the social foraging system, as we call it, which is a station where the birds can come to feed. This pyramid here sits on top of an electronic balance, and it's designed so that only a single bird can be accommodated on this little perch in the middle, and this is a slippy surface. That means we can't get several birds piling on there at once because they can't grip on. There's also an RFID aerial embedded in this pyramid, which can read RFID tags, which we put on leg rings mounted on the starlings. So the idea is that we can have one or more of these stations in an aviary. We can have birds, multiple birds wearing RFID tags, and we train them to come and perch on this little balance here in order to feed from a food hopper here. And then additionally, we have a pecking key here that we can light up in different colours, so we can actually run different kinds of operant tasks on this system. So we can ask the birds, for example, to peck the key a number of times in order to get a food reward from the hopper, which can be raised or lowered by a computer in the adjacent room. So this is a system for studying foraging behaviour and weight gain in the starlings, in social groups, without having to constantly catch them and weigh them. And this has completely changed the kind of data that I'm able to collect from these birds. So I think it's been both a welfare innovation and also a scientific innovation in that we can now get hundreds of body weights from a bird across the day and study how body weights responding to changes in the bird's environments, both social and food availability. So I just wanted to show you a little video of this because I'm quite proud of it. Aviary. They've got 2 social foraging systems. You can see the birds jumping up, pecking a key in order to earn food rewards. They can also explore on the floor, probing around in the substrate. They've got a water bath that they love to use, drinkers, ropes around the top that they can perch on when they're not foraging. So I think this is a much more welfare-friendly system for studying body weight and foraging in these birds compared to the previous kinds of systems that we were using in the past of putting the birds in individual cages and grabbing them to weigh them regularly. And I think modern technology means that these kinds of systems are now possible for all sorts of animals. So I think it's important that we really start to study body weight in a better way in the lab than we have been doing in the past. So I just wanted to show you some of the body weight data that we can get from this system. This graph here shows the mass data from a single bird on just one day. So what I'm doing is plotting time of day across the bottom and mass on the y-axis, and you can see all the dots of the individual stable masses recorded by the social foraging system. The data's a bit noisy because the birds jump up and down and they shit on the perch and all sorts of stuff happens. So it's not great data, but we get so many hundreds of points that we can fit it with a cubic polynomial in this case and estimate the body mass quite well over the day. And what you can see in this panel below here is daily trajectories for 12 birds estimated from this system. So this is based on, I think it was 10 or 11 days of data. So you can see what the daily mass gain trajectories look like for these 12 starlings in our aviary from this system. So that's the sort of data we can get from this system. And for the first time, this allows us to actually study the shapes of these daily mass gain trajectories in some detail. You can see that there is some variation here. And in fact, if you look at more data, you'll see even more variation, that a few birds show almost linear curves across the day, but for most, it's a non-linear curve with a more rapid gain earlier in the day and then a flattening off from sort of midday onwards. So the reason this is interesting is that the optimality models relating to body mass not only specify how average mass should respond to perceptions of starvation risk and predation risk in the environment, but they also make predictions about how the trajectory across the day should respond. So the idea is that if you're worried about starvation risk and food disappearing, you need to put on weight early in the day in order to protect yourself and have insurance against periods when you may not be able to forage. Whereas if you're more worried about predation, it makes sense to put off your mass gain until later in the day. You don't want to be flying around with excess mass because that makes you more vulnerable to predation. So you should actually put off your mass gain until later in the day and do it right before you go to sleep for the night. And there's lots of evidence that birds do change their daily mass gain trajectories according to risks that they perceive in their environment. So this is just one experiment on yellow hammers in this case, where they looked at body mass across the time of day, and they measured this both in safe conditions when there was no sparrowhawk present and in risky conditions where they flew a model of a stuffed sparrowhawk past the yellow hammers. And what you notice here is that this graph on the left is more linear, and the kind of point of inflection is further towards the left, whereas in this graph on the right, there's a sort of bit more of an uptick towards the end of the day, so it's a bit more kind of sigmoid in shape, and there's a steeper gain in the latter half of the day. And that's the kind of typical pattern you see when birds are worried about predation. So the reason I brought this up is that it led me to think that we might be able to use daily weight gain trajectories as a novel method for asking animals about their perceptions of threat in their environments, because there are lots of models predicting how these curves should look, and I wonder whether we could actually use this method for saying something about the different types of threats that birds are perceiving in their environment. So could we use the steepness of the weight gain curve early in the day and late in the day to say something about whether the birds are perceiving greater threat from starvation or predation. So just going back to our starling data, there's very little evidence in any of these for this uptick towards the end of the day, which had been seen in previous aviary studies of tits. And I wonder whether this is because we weren't actually having to grab the birds once an hour to weigh them, as some previous studies have done. So these birds were perhaps much less worried about predation than in some previous studies that have been done. Now, this is just speculation, but we do have the option with this new technology to actually test this idea that we could look at how worried birds are about, for example, being caught regularly in the aviary by looking at how they change their mass trajectories over the day. And that could be a way of actually getting a readout of their perception of threat of predation in the captive environment. Okay. So that's all I've got to say today. I need to thank Claire Andrews, who was our postdoc in Newcastle under the ERC grant that funded a lot of this work. And I talked to Claire a lot about these ideas, and she's got a paper coming out very shortly in Animal Welfare on the use of body mass measures in severity assessment in laboratory passerine birds, which I'd encourage anybody who's interested in these ideas to go and read. Thank you very much for listening.
Thank you very much, Melissa. If you have questions, please raise your hand, and if you can, go to the microphones. There are some on the side and in the middle. And if you could also say who you are and where you're from, that would be great. So we will start with you, Frank.
Hello, thank you. I'm Frank from the UFAW, Belgium. Very thought-provoking talk. Thank you very much. And actually, I have two questions. I don't know which one to ask first. But would you extrapolate your findings from wild birds to farming conditions, for example, broiler chickens, in which it is often assumed if they don't grow according to their potential, it is often taken as a welfare problem?
I think we need to think much more about strategic regulation and how it might relate to farm animals. I think some people are starting to do this, but it's really bringing together this literature from behavioural ecology that thinks about the function of fat with the welfare literature and also animal production literature. I mean, obviously, if we can make animals grow faster, that's a good thing for farmers. I think there's a lot of interesting ideas to be explored here that people are only just starting to think about. Yeah.
Thank you. May I ask the second question, or pass on to someone else?
No, Frank. The next one is Pernille. You were there. Where are you? There.
Hello, Pernille from World Animal Protection Denmark. Thank you for a very, very nice presentation and bringing us back to basis to what it's really all about, working with ornithology. I have a question regarding habituation. How do you, starlings, do they not habituate to a model of predators, and how will that influence their behavioral response and their weight loss or weight gain? And the second thing is that is there some kind of social habituation between a flock of starlings that will influence the individual birds?
So habituation to predator models. I mean, they probably would habituate to a stuffed model. When we have shown starlings stuffed sparrowhawks on occasions, we've only done it for very brief periods, so we've never done it for sort of hours on end, and they seem to respond really strongly to them. So, I mean, I think you just need to think about that in the context of whatever experiments you were doing and be aware that it could be an issue. I mean, it's not going to be very adaptive for animals to habituate to anything that might be a predator very fast, because it's a bad mistake to make. So I would imagine that habituation wouldn't be really quick. And habituation in the flock, I'm not quite sure. I mean,
I'm just thinking normally when animals live in a flock, I mean, like you said, some will be dominant and some will be subordinate, and that influences the flock and the response of the whole flock. Does that mechanism not occur in starlings?
Absolutely. I mean, I showed you Mark Witter and John Swaddle's experiment where they looked at the effects of dominance on body fat. And this is something that we're now actively pursuing with the social foraging system, because I can have groups of birds in the aviary together, and we can automate the recording of dominance using the social foraging system, because when the RFID of one bird is rapidly replaced by the RFID of another bird, we can count that as a dominance interaction, and we can validate that with video evidence, and we've done that. So we can now use that RFID data to get real-time estimates of dominance relationships in the aviary and look in quite kind of on a fine time scale how that relates to changes in body mass in the birds. So that's something I'm actively pursuing at the moment, and I think it's really interesting to know how the social relationships in a group alter body mass. And that has real implications for if you're moving animals around in the lab, if you're changing the composition of groups, for example. You might need to take account of the fact that changes in body mass might be due to changes in dominance that are happening. So if a bird suddenly loses weight, it might not be because there's anything wrong with it, it's just because it's suddenly the dominant bird in that situation.
Siobhan.
Thank you. I wondered if I'm right at the top. I wondered if you could comment a bit on hunger and the experience of hunger, and whether you have any insights into that. I'm thinking particularly of, many of our farmed animal species are so highly motivated to eat. Sometimes we seem to be providing them sufficient food, sometimes they're not, sometimes they're losing weight, and kind of hunger is a natural homeostatic mechanism that can tip over into a very negative experience.
Yeah, I don't know about that really. I mean, I suppose one of the really interesting things that's come out of our recent research is that we always assumed that the fat gain in these birds that were food insecure was due to them eating more. And as I mentioned, one of the things that's come out is that it's not, and that the birds don't eat more. They manage to gain weight without eating more, and in fact, in many of our experiments, they actually eat less overall. So food insecurity is not apparently increasing the hunger in the birds. What it seems to be doing is having effects on their energy expenditure, so how much they move around possibly, but also very likely on their metabolic rate. And I think energy, as I said, is being shifted from somatic maintenance and repair into fat. So there's some really interesting biology going on there. How it all relates to hunger, I don't think those food insecure birds are necessarily more hungry. Although, they do show faster foraging and they eat faster when the food is there. So there was some evidence that they pecked the key more and they ate faster. So there was some evidence that their hunger might have been higher, but they didn't eat more overall, possibly because of the food insecurity. I don't know.
Fine, thank you.
And a last question here from Georgia.
That was so fascinating, Melissa, and I'm wondering if you know whether there's work on how animals strategically store their body reserves. So, for example, where they store their fat. In humans, it might be on your bum or your belly. Is that strategic? And also whether you store surplus energy as fat or glycogen. Are there kind of optimal rules that determine that?
Probably, but I'm not sure that I can say anything intelligent about it. at this moment. Yeah, I mean, there's lots known about different fat storage in humans. And sort of visceral fat is very different from subcutaneous fat. And in the birds, we don't know that much about it. The only, when we've looked at this furcular fat store, which is the one that's kind of in the throat here, that you can actually see externally. So that's something that comes and goes very, very rapidly. In little birds, you can actually almost see it come and go by the hour. If you look at them, you just blow away the feathers and you can see this yellow shape that gets bigger and smaller with time, which is amazing. So that's, I mean, fat that's definitely being strategically regulated over the day to support normal life. But yeah, I don't know about switching between different fat stores. I haven't really done any work with that. It's not something we can get at terribly easily in the birds. I have been exploring putting them into a micro CT scanner to see if we can actually look at where the fat is and how it changes over time, but that's quite difficult because it's shared with, it's mostly used by rodent people who work on animals that might have compromised immune systems, and putting wild birds into it is, we're going to get there, but it's going to take a little bit of time, I think.
Right, good luck.
Thank you very much, Melissa. Please help me give Melissa a big round of applause. So our next speaker today is Professor Alistair Lawrence. He is a professor at SRUC at the University of Edinburgh. He is also my old PhD supervisor. Take it away. I didn't mean to say old.
Previous. Good morning. Brain health, and time to justify this title. What is brain health? Well, it's a lot to do with the mechanisms that underlie brain homeostasis, as illustrated in this definition by Gorlick et al. And most of what we know about brain health comes from animal studies, translational science. And the whole point of what I'm going to talk about today is that the concept of brain health has really not been applied to animals in their own right. And this talk is going to try briefly and hopefully successfully to illustrate some of the value of looking at animal welfare through the lens of brain health. Now, there have been some papers out, reviews, which have looked at neuronal biology and its relationship to animal welfare. So in the first part of this talk, I'm going to look at non-neuronal constituents of brain health. So the human brain is about half neurons and half other cells. And those other cells are mainly glia, or glial cells. Previously thought to be inert, glue, but now known, as we'll see, to have a number of really functional importances to brain homeostasis. Now, this ratio of glia to neuron actually varies across species. For reasons which are not yet clear, it could be, for example, due to neuronal size across species. The rat, as it happens, has a glia to neuron ratio approximately the same as a human. So this drawing shows some of the cell types in brain tissue. And I'm highlighting here astrocytes. They constitute 20 to 40% of glial cells. They share a common origin to neurons, but they also have immune-like functions. For example, they can secrete cytokines. And as this drawing shows, they interact very closely with both neurons and blood vessels. So what you're seeing here is a depiction of what is sometimes called the tripartite synapse. And it's really a close physical relationship between astrocytes and synapses, which has been argued to help the brain achieve functions across spatial and temporal dimensions. Did Einstein owe his genius to his astrocytes? Well, it was the only difference, apparently, in his brain, that he had more astrocytes in certain regions of his brain. And certainly astrocytes seem to have something to do with learning and memory. This very recent paper, which decoupled astrocytic networks, showed that that had important impacts on learning and memory. The rather remarkable idea of engrafting human astrocytes, which are more complex, into neonatal mouse brains showed that when these mice matured into adults, they had sharply enhanced learning and memory. Slightly freaky. Microglia. These constitute maybe 10% of glial cells in the brain. They are the brain's immune cells. They are macrophages, and hence they have a different origin to astrocytes and to neurons. Now, you might think that microglia are there for immune purposes, and they are, but what has become apparent in recent times is that they also have a number of important homeostatic functions. For example, synaptogenesis and synaptic pruning. And they also appear to have something to do with psychological functions, such as learning and memory, perhaps through the release of physiological levels of inflammatory cytokines. Now, both astrocytes and microglia respond to challenges such as stroke by becoming pro-inflammatory or reactive. And this change to a more pro-inflammatory state is accompanied by changes in their morphology. So this slide taken from Neil Mabbott's lab shows astrocytes stained with GFAP, which is an astrocytic marker. They're reactive. They're in the brain of a mouse going through prion disease. And similar morphological changes are found in microglia, as shown in this depiction here from Carperian. You can see the microglia moving between a ramified or homeostatic state through to what is variously described as reactive or amoeboid or sometimes just blobby. And these morphological changes are accompanied by gene expression changes, which are pro-inflammatory in nature. And in some contexts, these reactive glial cells are thought to become part of the disease process. They make things worse. For example, possibly in human depression and chronic anxiety. So I want to move on to another constituent of brain health, and these are the barriers that separate brain tissue from blood. These physical barriers prevent soluble molecules, well, they don't prevent, but they restrict soluble molecules entering brain tissue. They contribute towards what's called the brain's immunological privilege. And they probably function to prevent or to try and control inflammation in the brain. We have a hard skull, and inflammation is not a good thing. And the best known of these, of course, is the blood-brain barrier. And it's depicted here, and you can see astrocytes are involved again. So we're talking about the cerebral microvasculature, which occupies a significant proportion of the volume of the brain. And this drawing is a cross-section through a brain capillary, and it's showing what is referred to as the neurovascular unit, which is a modular arrangement of different cell types, which again, main purpose of is to restrict soluble molecules entering brain tissue. And I'm just going to illustrate two cell types here, endothelial cells, which form tight junctions, and again, the astrocytes. And astrocytic end feet form an important contribution to the NVU. And this is a recent high-resolution electron microscopy, just to show that there's some evidence for these drawings, showing exactly the structure which I've just described with the endothelial cell tight junctions and the astrocyte end feet, as around the capillary. But there are other interfaces between blood and brain tissue. For example, in the meninges. And there is quite a lot of interest in the immune cells which occupy these meningeal spaces. So this is a cross-section through meningeal tissue. The dura is rich in immune cells. I'm just showing here meningeal T cells as an example, because as an example, they've been shown to be involved in learning and memory. And a recent study, again, showing that a subpopulation of these meningeal T cells is involved in anxiety, although the mechanism is as yet unclear. Now, physical disease has been known for some time, like stroke, to affect the integrity of the blood-brain barrier. And it's now becoming clear that other challenges, including psychosocial stress, can have similar effects. So what we're showing here is neuroinflammation activating endothelial cells, which then allows immune cells, which shouldn't be getting through, into brain tissue. And that very passage of these immune cells through the endothelial cells is then thought to activate the astrocytes, which may then further reduce the integrity of the blood-brain barrier. So what is the relevance of this to animal welfare? So the rest of the talk is going to look at 3 examples where there is, I believe, relevance. And I'm going to start with sickness behaviour. Sickness behaviour is a well-described process where peripheral disease or infection leads to perturbation of brain homeostasis, which then affects psychological functioning and welfare. It starts with a pathogen, a virus say, which is recognized by the peripheral immune system, and those immune signals are then transduced to the brain through circulation, but also through afferent nerves. And the end result of that is the production of pro-inflammatory molecules, including cytokines, by microglial cells. And this picture from Rod Johnson's lab shows activated microglia, reactive microglia, in the brain of a pig infected with PRRS virus. And the behavioral sequelae of this are well known. Motivational reorganization, anorexia, decreased social behavior, and exploration and lethargy. And there's a lot of interest in sickness behavior, for example, as an early indicator of disease in farm animals. There's also an interesting recent hypothesis that sickness may be an important cause of damaging behavior such as tail biting or ear biting in pigs. And there has been some interest, some exploration of the experiential component of sickness behavior, what it feels like to be sick as an animal. I would say this is an area which does require more attention. My second example is social stress. Chronic social stress is a widely used model in translational science. It almost always involves variations of the resident intruder test, often over multiple sessions over days. And this social stress is believed to activate threat appraisal circuitry in the brain, which in turn activates peripheral stress pathways, the HPA, the sympathetic nervous system. And one peripheral consequence of this is to prime immune cells in bone marrow. And these primed monocytes are then believed to traffic back into various tissues, including the brain. So the general hypothesis here is that these primed monocytes entering brain tissue cause neuroinflammation and chronic anxiety. So again, we're seeing this cross-section through the brain capillary. We're seeing reactive endothelial cells facilitating the trafficking of these monocytes into brain tissue. We're seeing these monocytes most likely differentiating into macrophages once they're in brain tissue. And this flow cytometry plot shows what's at least evidence of the accumulation of these monocytes or macrophages in a mouse brain following social stress. And the welfare endpoint, I suppose, is that these now brain macrophages, but which were monocytes, appear to enhance, maintain the threat circuitry and potentially lead to a state of chronic anxiety. And the relevance, well, social stress is one of the most common consequences of the way that we manage our animals. And understanding the effects of social stress on brain health could at least, for example, lead to a greater appreciation of the consequences of social stress, for example, chronic anxiety, and potentially also novel interventions to reduce the consequences of social stress or the impacts. I'm being flashed here. Keep going. Environmental enrichment is my third example. Again, widely used in translational science to study the effects of environmental complexity on brain development and brain function, with many reported effects on the components of brain health I've just been describing. Environmental enrichment in rodents is often associated with anti-inflammatory profiles, gene expression profiles, or physiological profiles. The clearest effects of these involve physical exercise. Novelty is less studied. Other aspects such as novelty are less studied, but they appear to have similar effects. In the pig, we've previously published research showing that enrichment in pigs appears to have anti-inflammatory effects in the brain. What we found was genes, microglial genes, downregulated, associating with the behaviour which was stimulated by the enrichment. Now, for this work, we had to refer back to human and mouse gene lists, which are called microglial gene signature lists. And as an advance forward, we just published this paper, which is a microglial gene list for pigs. We've used that here, for example, to look at the comparison between gene expression profiles across species, mouse, human, and pig. And it will in the future help us, for example, develop new markers for microglia in pigs. So what's the relevance? Well, of course, we know, we've had many talks about it here, domestic animals tend to be kept in environments that restrict exercise and exposure to novelty. I think the rodent enrichment work, again, raises really interesting questions about what is true enrichment. And I want to, again, refer to the work that Baz referred to yesterday. I think the bidirectional relationship between brain and the immune system is really worthy of more attention, particularly in the context of what enrichment might be doing to enhance disease resistance. So my summary, there's a wealth of knowledge out there relating to brain homeostasis, mostly conducted in translational science. There are a number of examples of how brain homeostasis can break down with relevance to animal welfare. These examples, I think, help to show the relevance of brain health to animal welfare, and we think there's great potential in developing this area to the improvement of animal welfare, and there's a written review on its way, if somebody could just get a move on. And I would like to acknowledge my co-workers, co-authors, and right now I'd like to pay particular homage to Taylor Hammond, who, despite the stress of trying to finish off her PhD, took the time off to do the drawings for this presentation. Thank you very much.
Thank you, Alistair. He has cunningly gone so much over time, there's only time for one question. If anybody would like to, yes?
Hi, Alistair. Thank you for a brilliant talk. Fascinating, really. I just wonder how to assess brain health non-invasively.
Non-invasively. At the moment, of course, MRI, if you count that as, or imaging in general, MRI, PET scanning, all are opportunities to look at the brain. Of course, you then have to validate that, and we're doing that at the moment. We've got MRI images suggesting effects of environment on different brain regions in pigs. We're now looking at what that might mean in terms of the actual tissue itself, what is actually changing to cause these changes within the MRI image. Thanks.
So should we now, as an animal welfare scientist, should I then now collaborate more with neuroscientists, or what's the way forward?
I knew this question was going to come up. I guess it's not for everyone, but I certainly think the experience I've had of reading into this literature is it is vast, it's incredibly fast growing, but it's fascinating. I mean, it just attracts your interest, and if you can find a friendly neuroscientist who has the time, then yeah, I would definitely engage. And I mean, what they need, I think, and we've been talking about this a lot at this meeting, is relevant models. You know, they do a lot of it in mouse models or other model species, and if you can bring to them a species like the pig, they begin to get quite interested. So that's been one of the reasons we've been working with them.
Thank you very much. Before I hand over to our next speaker, I'll just say that since yesterday we've had about 40, 50, 60 people more signing up to the online transmission. So welcome to all of you who are listening from your own office or room. Our next speaker today is Matthew Craven from Newcastle University, who will talk about complexity of aviary design.
Good morning, everyone. So this morning I'm going to be presenting my most recent study from my PhD project, which I've recently just completed this part of. So in the UK, the cage-free systems are becoming much more popular compared to cage systems for laying hens. With cage-free systems, including free-range, which is the majority, organic and barn systems, making up almost 2/3 of the total UK egg market at the moment. So this proves very popular with UK consumers. But within cage-free, and even just within free-range systems, which is what I'm focusing on for my project, there are a variety of different housing designs which are in use, which can vary quite a lot in complexity. So this study is going to compare two of those examples. The first of these is a flat deck system, where you can see here we have a large flat floor with a raised area that gives some opportunity for perching. And the other system is a multi-tier, where we still have a flat floor level, but this is much smaller, and there are lots of racks. I think it was 5 different levels were available to the birds in the system that I studied. So this provides a lot more opportunity for exploring the 3-dimensional space within the aviary, which we believe gives the birds more spatial stimulation. And I was interested in testing the differences between these two systems in terms of both spatial stimulation for the birds, and to see whether this had an impact on their welfare. So the measure of welfare that I used during the study was hippocampal plasticity, which is where we have these immature neurons which are changing in morphology, they're migrating and being integrated into the hippocampal network. And an extreme form of hippocampal plasticity would be neurogenesis, where we're seeing newborn cells being added to the hippocampus. There's been a lot of research on this, mainly in humans and in rodents, and in humans suffering from major depressive disorder, neurogenesis is known to be downregulated. There have also been many studies in rodent models of other depression-like states, so not just in depression, but also chronic stress, chronic pain. We see fewer of these immature neurons, decreased hippocampal plasticity. However, positive experiences and treatment with antidepressant drugs, and also positive experiences such as environmental enrichment and exercise, have been shown to increase hippocampal plasticity. So like I say, most of this work previously has been done in rodent models. So the main regions that we're interested in are the dorsal dentate gyrus of the hippocampus in rodents, which is where we see a role of hippocampal neurogenesis in spatial memories here. But we have a functional gradient where in the ventral dentate gyrus, we see that that hippocampal neurogenesis in this part of the hippocampus is more sensitive to emotional stimuli. So this is where we would see effects of chronic stress and other depression-like states. So why is this a useful tool for people who are working in poultry welfare if most of the previous work has been done in rodents? The morphology of the avian hippocampal formation is very different to what we see in a mammal. So here on the left we've got a rodent, this is the dorsal dentate gyrus in a rodent brain, this green line here. And in the avian brain, this green line here, the V-shaped region, is believed to have a similar role to the dentate gyrus. And though these look completely different from each other in morphology, that functional gradient appears to be conserved. So here we have an avian brain, and it is the rostral hippocampal formation here at the front of the brain. It sits kind of here down the midline, the medial part of each hemisphere. That is the part which has a role in spatial memory, and the caudal HF at the back of each hemisphere of the forebrain here in red. This is the part which is sensitive to emotional stimuli. So since my study is interested in both spatial complexity and stress, here we're interested in both of these subregions. In our lab at Newcastle, we've previously validated hippocampal plasticity as a biomarker of chronic stress in birds, specifically laying hens. So when subjected to conditions such as feed restriction, unpredictable chronic mild stress, and keel bone fractures, which are very commonly experienced by laying hens, we see reduced hippocampal plasticity at the caudal pole of the hippocampus. So for my study, I'm planning to build on this previous research, and my first hypothesis being that hens housed in more complex aviaries experience higher spatial stimulation than those housed in less complex systems. If true, we would predict that multi-tier birds, so the more complex of the two systems I'm looking at, will show increased hippocampal plasticity in the rostral subregion of the hippocampus compared to flat-deck birds. My second hypothesis, which is the one which is more about welfare, is that hens housed in more complex aviaries experience lower levels of chronic stress than those housed in the less complex system. And again, we would predict that multi-tier birds here would show increased hippocampal plasticity, but in the caudal subregion of the hippocampus this time. So my methods for testing these hypotheses started with a flock of birds who were all within the same rearing shed. At 14 weeks of age, we sampled 12 of these birds from that rearing shed and sampled their brains. And then 2 weeks later, the birds from that rearing shed were split into two laying barns. One of these was a multi-tier and one was a flat deck. After 8 weeks of being stored, I've got out of order there. The multi-tier and the flat deck were both managed by the same egg company, and they were as close together as we could get them geographically to try and control the two systems as much as we possibly could. So after the birds had been settled into these two adult systems for 8 weeks, we sampled 11 birds from each system, and again, in the same way as we did with the pullets, we sampled their brains. So on the days that they were taken, we would weigh the birds, we would weigh the spleen of the birds as well and sample their brains, which were then fixed and cut to coronal sections and stained to show up double cortin expressing cells. So double cortin is the marker of plasticity that I used in this study. So that is expressed only in cells which are considered to be plastic, so they are the ones which are migrating and still becoming integrated into the network. After the sections were stained, I selected 5 rostral sections and only my one biggest caudal section, because there aren't as many of them. It's just the sheet which kind of folds over the back of that forebrain, which I showed you before. And for each one of my sections, I would draw around the hippocampal formation here, and I would visit hundreds of sampling sites within the hippocampus and work through those systematically to count these double cortin expressing cells. So for each site, we would magnify to 100 times magnification, and this is what those cells look like. So these darker cells which are standing out against the lighter background are the ones which are expressing double cortin, so they're believed to be the ones which are new and immature. I was looking at 2 different kinds of these double cortin expressing cells. So on the left here we have the bipolar ones, which are the younger cells. They have this fusiform shape with 2 processes. And we have the multipolar cells, which tend to be larger and have more processes, and they are still immature, but they are slightly more developed than the bipolar ones. So now that we know exactly what I'm looking for plasticity, I'll just revisit those 2 predictions of the study, which are if we've got a higher environmental complexity in one shed than the other, this provides greater stimulation, the multi-tier birds will show a higher density of these DCX cells in the rostral HF. And also, if we've got a higher environmental complexity and that causes lower chronic stress, the multi-tier birds will show a higher density of double cortin cells in the caudal HF as well. So moving on to my results, I'll start with the body mass because we weighed the birds before taking their brains. We do see a difference in body mass here, but that's not surprising because the pullets were obviously younger and smaller than the birds taken from the adult aviaries, so that was very much expected. What is most important from this slide is that the flat deck and the multi-tier birds, which were the same age, didn't differ in body mass. And also, after correcting for body mass, here we have the residual spleen mass, and there was no significant difference here between any of those groups, which suggests no difference in immune function or disease status between those groups. So now moving on to the most interesting part, which is the results from the brain. These were analysed using a linear mixed model with housing group as our between subject factor and cell type and subregion of the hippocampus as within subject factors. And I found no significant main effect of housing group, which was unexpected. I did find a significant effect of cell type with more bipolar cells than multipolar cells in all groups. I'll point out as well that these are Z-scored to standardise between staining batches. That's why we have some negative ones. But yeah, there were significantly more bipolar cells than multipolar cells in all cases. And also there was a higher density of both cells in the caudal hippocampus than the rostral. So if I take a look at each of those subregions individually, starting with the rostral HF, which is the one which is sensitive to spatial stimulation. This is the same data, and we see there is no difference in bipolar or multipolar cell density between those groups, which suggests no significant difference in spatial stimulation when moved from the rearing site to either of those adult laying systems. And in the caudal hippocampus, we see a very similar story with no significant differences between any of these groups, which suggests no difference in chronic stress experienced by the birds in each housing group. So in conclusion, the difference in complexity between the flat deck and multi-tier aviary was not great enough to have a detectable effect on hippocampal plasticity. So there may not have been any real difference in complexity between those systems at all, or at least it wasn't severe enough to cause any differences in the hippocampus. Also, because we didn't see differences in the caudal HF, which is sensitive to stress, the results suggest that any differences in welfare between these two aviaries would be minimal. And while there may be alternative reasons why you would choose a multi-tier over a flat deck, for example, more efficient use of the floor space, farmers are not introducing a source of chronic stress if they continue to use a more conventional flat deck rather than switching to those multi-tier systems. So I'd like to thank you for listening. I'd like to thank my supervisors and past and present members of Smulders Lab at Newcastle who have done the work leading up to this. I'd also like to thank the staff at the farms who helped me with selecting the birds. And any questions?
Thank you very much. Are there any questions for Matthew? We have two here. I'll start in the back. Yes, Heather.
Hi, Heather Browning from London School of Economics. I'm really interested in this idea of hippocampal plasticity as a welfare measure. But what I'm wondering, is there any way of performing the measure on animals while they're still alive so we can get a sense of their welfare throughout their life, or is it only post-mortem?
Currently, it's only post-mortem, as I know, which is inconvenient because we have to kill the animals, and then it also takes a very long time to process the brains once we've extracted them. So if there could be a way to do this in live animals, that would be much more convenient and much more useful for those animals, yeah. But no, currently not available.
So currently there's no work that you know of to try and develop this, or?
Not that I'm aware of, no.
Okay,
Sorry.
thank you.
Hello. Thank you. I'm Julia from University of British Columbia. Thank you very much for your presentation. I just want to ask, because I saw the last slide, it's a free range system. Do you think the range access has any kind of potential consequences on it? So for example, if it's just indoor only, would that also have a difference? Yeah.
I don't have data to support that because I've only tested animals who both had access to an outdoor range. So I don't know. Well, my results didn't point to a difference between the complexity, but I would expect that a bird which has access to an outdoor range would have more spatial stimulation than one that doesn't. So it's possible.
Buzz.
Thanks, Matt, for an interesting talk. I was wondering about the length of time you put them in the adult system, because it's only 8 weeks, and it's also the period that they have to adjust to the new system. So would you expect different results maybe a bit further in the laying period, once maybe also the difference in lifestyle with migrating through the system is more internalized by the birds?
Possibly. So we did actually take a second sample. I think it was another 24 weeks later. So once they'd had much more time in their laying sheds. And unfortunately, I won't have time to analyse those brains as part of my PhD. But the reason we took those was to see if that extended time made a difference. Previous studies that I cited there have only used 8 weeks of chronic stress, but those seem to be much more severe stresses, such as keelbone damage, which did make a difference.
I have one there.
Hi, Pernille from World Animal Protection Denmark. I was just wondering, now that you did sample the brains and killed the bird, did you look at the state of the keelbone? Do you have any records of the level of keelbone fractures in the two housing systems?
So for the birds that we sampled, we only took birds which did not have keelbone damage because we know that that has an effect on neurogenesis. We wanted to just look at the systems.
How do you know if they have keelbone fractures looking at a live bird?
You can just palpate them, so you can hold them against your chest and feel along the length of the keelbone. You can be trained to feel for any deviations or fractures.
So it's not differences in behaviour?
No, we didn't do any behavioural measurements for this study
Thank
either.
You.
John Amory, Riddle University College. I was interested, you had 18 weeks in a rearing accommodation. I wondered if we could have details of what the rearing accommodation was and how that was. It multi-tiered? Did they have access to purchase in the rearing accommodation? And does that fix any of the plasticity before you would even start the trial?
So it was mostly flat with some access to raised perches when the birds were large enough to use them. So they would be introduced as day-old chicks and they would just have the floor space. Then as they grow, sort of tables are let down which they can then perch on. Yeah, that's interesting because the amount of time that they spend in that environment, they will be subject to those new cognitive challenges, so there will be changes in plasticity during that time as well. I'm not sure how much of an effect that would have had later in life.
Thank you.
Thank you.
Thank you very much. And we have come to the last speaker this morning's session, which is one of the two winners of the UFAW Early Career Researcher of the Year Award. Nienke Van Staaveren will talk about feathers, feet and fractures.
Thank you. For those of you who are wondering about this title, this was the only way I could get the alliteration to work. Bones didn't really fit. But it also is touching up on a lot of the topics I want to cover today. And on first glance, when you look at these sort of things, that you might not see the connection that clearly, but if we talk about issues like feather damage, footpad dermatitis, keel fractures, it's clear that those are all important welfare issues. And in part, that is because these are what we can consider different forms of injuries if we go by this definition that it's any form of a disturbance or damage to either the structure or the function of any part of the body. My name is Nienke Van Staaveren. I am a senior research associate at the University of Guelph. And the past couple of years, I've been looking at these different types of injuries in different contexts. And doing that, I've come to realize we're often very much focused on one particular topic, very much zooming in on one piece of the puzzle. But by doing so, sometimes we tend to forget that it's part of a whole animal within a system. So it's a lot more complex. And with this presentation, I want to highlight a little bit of those findings that we've been seeing that coming together. So research, when we're zooming in on that one piece of the puzzle, in reality, as I said, it is more complex. We're seeing issues like co-occurrence of different diseases. Comorbidities is a word that's often thrown around these days, but also things such as muscle myopathies. But when it comes to welfare issues or these injuries in particular, there's not a lot of data out there if they are actually co-occurring or how that might impact our poultry and their welfare. There are a few reports, especially in meat birds or turkeys, as an example here, that there is potential for a sort of vicious circle, if you will. So there's a few reports that birds with footpad dermatitis, for example, also have other forms of contact dermatitis because they're lying down more, or they may be more likely to get pecked at because they're not as mobile. But a lot of that is more anecdotal, or only a few reports that are actually supporting it with data. If you try to find actual numbers, we couldn't at the time that we were looking into this. So we had the opportunity to actually add a little bit to that as part of a larger project where we had been looking at different injuries in turkeys. So we were looking at injurious pecking, so injuries to the head, neck area as a sign of aggressive pecking, but also the back tail area as a sign of more feather pecking, as well as footpad dermatitis. And we had a lot of data on a lot of these birds, and we could actually tell, okay, which injuries or how many birds are having those different types of injuries at the same time, which was a very simple check, but thankfully not many of them had more than one injury at the same time. Most of them just had one, but still about 7% was showing with more than one injury. 7% may not sound like an awful lot, but if you consider the size of the flocks or the size of the sector, it could potentially still impact quite a large number of birds and potential have welfare implications if we assume that having more injuries is worse. And we also broke this down to see which combinations are most frequent. So that was the combination of injuries to the back as well as footbed dermatitis. That was clearly the most common, but also doing some analysis here, that one and the different pecking injuries, they were actually statistically also associated with one another. And this is important to realize, especially for farmers, but we worked with them a lot, that they just also realize that you should focus on multiple things at the same time. Don't get distracted by the one visible injury, for example, which can happen with the back or the pecking injuries. Those are often more clearly visible than something like footbed dermatitis. And this was one aspect that we were looking at, but switching gears a little bit, apart from co-occurrence, there's also some other implications of injuries, and for that we're just going to talk on the laying hens here, because here the story of the keel bone damage comes back into play. And this is really work that's been very much developed by Dr. Alexander Harlander at the University of Guelph, together with a very large team. It started before I arrived, so not everybody's shown here, but I do want to point out especially our collaborators in the US, Dr. Don Powers and Dr. Brett Tobalska, who are experts in avian flight and energy expenditure, but mostly in wild species. And that's really interesting for us because we've been trying to adapt some of their knowledge, some of their techniques to our laying hens, which is not always as easy, it's quite complex. Trying to do this in research barns, but we're trying to answer questions like, what influence do different injuries have on their locomotion? Does the use or disuse of muscles influence the development of keel bone damage? And as that research program has been building, we're starting to see some connections that I want to highlight now. And one of the first things that they've been looking at is actually the influence of those injuries. And what I want to touch on now is the landing, especially because that's a behavior where birds very much rely on their wings and on their legs to either break and absorb the landing. And you can imagine if birds have footbed dermatitis or if they have keel fractures, that might not go as smoothly. And we've been doing that by training birds to land on force plates, first from a 30 centimeter drop, just so that they know how to land properly, and then from increasing heights. So for that, we've been using this jump tower. You see Raul here, who is one of our undergraduate students. He was really the first to train these birds on a lot of these tasks, which was very great for me because he could actually reach the top of the platform. And this is what that looks like of her feet in that designated square. This worked pretty well, but probably most of you who have been working with live animals and technology doesn't always go the way you hope when you actually need it to. So we had hoped to train up an equal number of birds that were considered healthy or that had footbed dermatitis or keel fractures. And so we didn't get the full data for all of these simply because some birds didn't land correctly or we had some issues with the force plate itself. But from those that we did have data, we extracted information, and from that we could calculate things like the landing velocity, the maximum resultant force, and the impulse, which is the area under the curve, which tells you something about how stiff-legged they're landing or if they're bending their knees to absorb the force. And this is data that we're still very much in the process of analyzing and interpreting, so I'm only going to show some very preliminary results from the 30 centimeter drop. And the interesting thing here is that actually we didn't see a difference between our healthy and our injured birds when it came to the landing velocity. The difference was actually between the birds with footbed dermatitis and keel fractures. So those birds with keel fractures were landing with a somewhat higher landing velocity than those with footpad dermatitis. We still have to validate this further also with our higher jumps, but the idea what we are having here is that potentially those birds with footpad dermatitis are somehow changing the way they land so that they don't have as much pressure on their feet. And the birds with keel fractures, they may be more hesitant or more reluctant to use their wings when they're approaching the force plates because those wing muscles are attached to that fractured bone. As I said, we still have to validate this, but this work, as well as some work that we published last year, it's giving us a general story that these birds are not very good when it comes to landing or flights. So they're landing at velocities that are about 2 to 3 times higher of more agile flyers, such as finches or doves. But actually, in this particular case, it's very similar to the speed with which human children land. And human children aren't really designed to fly. So it's a little bit concerning for our laying hens. But that's something that's quite interesting because they do need to fly. They're more ground-dwelling species, but they do need, especially in our housing systems, to be able to move up and down. And for that, they also use their flight feathers a lot, also for other things, but in the context of this talk, focusing on flight. And if for any reason the flight feathers are damaged, which can happen because of feather pecking, but also abrasions or any other sort of reason, you decrease the wing area. And when you decrease the wing area, you increase wing loading. So basically the bird has to support the same amount of weight, but they have a smaller wing surface area to support that. And that could have implications for power and their maneuverability. And so we recently, I think earlier this month, published one of our last papers where we actually tried to look into this by manipulating the flight feathers ourselves. So we had a control group with intact flight feathers and a group where we had the primary flight feathers clipped and also a fully clipped group where both primary and secondary flight feathers were clipped. And those birds were housed in floor systems, but they had elevated platforms, and they had different individual resources, so feeders and nest boxes, both on the ground and on those platforms. And those were equipped with RFID technology, so we could see which bird was spending how much time where. And that gave us a very clear picture of what's happening behavior-wise in these birds when you manipulate the flight feathers. And that is shown here. So this is how much time they spent at the elevated or the ground feeder at week 0, which is right before we applied our clipping treatment. And over time, that was a very clear pattern with our fully clipped birds. They consistently spent less time at the elevated feeders. The half-clipped birds were still able to get up, so that was good, but once you clipped both primary and secondary, they couldn't anymore. We also then looked at measures of the muscle and the keel bone itself. And for the muscle, we used ultrasound techniques and keel bone we did by X-ray. And that result, for the muscles, we were using the thickness of the pectoralis muscle because we didn't want to sacrifice our birds because we wanted to follow them over time. And the thickness is correlated with the size. So interestingly, what we saw was nothing was really happening with our control birds, but the half and the fully clipped birds, they were seeing a reduction of about 6% in the muscle thickness. So that combined with the changes in wing areas, probably what's leading to the changes in behavior. When it comes to the keel fractures, we didn't really find any results there, no impact, but also because we didn't have a lot of severe keel fractures. It was quite mild throughout the study, and it stayed very equal from before to after clipping. So that's quite interesting on its own. So I'm supposed to be wrapping up, which is perfect. So just to wrap up some take-home messages, I think it's clear we're starting to slowly fill in some of the pieces of the puzzle, but there's still a lot to be done, and a lot of our students are actually still working on this. But I think it's important to raise awareness that those different types of injuries can co-occur, and potentially they can worsen each other, which is something to keep an eye on. But also, apart from being welfare issues on their own, it's important to keep in mind that they can influence behavior and physiology in other ways as well. So, especially when we're talking to farmers right now, even if we don't have the full picture, we are trying to really hit home that message that you need all those pieces of the puzzle aligned to make sure that we have birds that are very well suited to the housing systems. And with that, I'll just thank all of our fundings and UFAW especially as well for bringing me here. And I'll be happy to take any questions.
Yes, Melissa.
Thanks for a very interesting talk. I was wondering about whether there's possibility of bias in your landing velocity results, and that from the birds that wouldn't learn the task. So you seem to have many fewer in your
with the injuries.
In the injured groups that learned the task.
So from what I remember, some of the birds especially with foot pediclitis, were hesitant to jump. So they actually trained up a few extra to bump up those numbers. With the data that we had in the end, we had more or less equal groups, but the groups were quite small. So
but the ones that didn't learn the task might have been the ones that really didn't want to jump off a high platform because,
Exactly.
I don't know how you deal with that, but it seems to be a concern that you might be underestimating the effects.
Yeah.
I have a question. In the fully clipped wings that you had, you saw a reduction in the use of the high feeders. Was that because there was only a few of those birds that could do it, or was it a reduction in all of the birds, so they just did it less?
I think in general it was a reduction in the fully clipped birds. The others still went up.
Yeah, but among the fully clipped birds, there were still some. They were still using the high feeder for 25%.
You mean like individual birds specifically?
Sorry.
Actually, I am not sure because we didn't have that many within one pen. But we should be able to trace individuals. But we just went with a group. But yeah, and I think that, and as well something that we didn't really check or couldn't really analyze yet, but is interesting, is also competition or individual preferences from the start.
Yep. Any more questions? Yes, we have one here.
Maeva from Utrecht University. You mentioned you had brown and white hens. Did you find any difference there?
Yes, the brown hens were definitely more on the ground. So we didn't see any change for them in terms of accessing the elevated feeder, simply because from the start they were feeding on the ground. We did see a reduction for them going up for the nest box. And they're also in the half-clipped groups. So it seemed like the change in the wing area there was actually having a bit of a stronger effect once they do want to go up.
Okay, thanks.
Thank you.
And a question from up here.
Yeah, hi, Pernille. Wildlife Protection Denmark, as you might have guessed, I used to do science in poultry. That's why I'm asking all these questions. And I have a question of clarification, because when you say you assess keel bone and fractures, do you distinguish between the fractures that can be touched, on the outer, that can be by palpation, or the inner fractures that can only be, you only see if you kill the bird or put them into a CT scanner?
So what they did do palpation, but the data that we used are all from X-rays. So they took X-rays of the birds and then scored from those scans.
So what do you find? I mean, the fractures you see, are they because of damages when the birds flew into equipment, or do you find the fractures on the inner side of the keel bone?
I think the majority of them were on the caudal tip, but what the actual cause is of those, I think that's the million-dollar question.
No, okay, yeah, okay, thank you.
Thank you very much. Before I let you all go for a break, if any of you have luggage that you want stored, you can put it in the cloak room just around the corner out here, but please don't put them on the stairs because that sort of prevents people from moving back and forth. And with that, thank you for attending, and see you back here at 11:20.